Hello there, Global Bioimaging and Exchange of Experiences. I'm really deeply honored to be here today, albeit virtual. And I'm going to make every effort, by the way, to try to be there in person in Montreal next year. I heard it's in Montreal. So, yeah, my name is Ryan. I'm in California. And I'm part of the NASA Open Science Data Repository. And I'm excited to present to you. I'm going to be talking about our repository, its capabilities, and especially bioimaging, of course, and as part of today's presentation. So several of you might be asking, why are we even here? I'm going to go through sort of the science question just quickly. When people go to space, humans and biology, as we try to make new homes for humanity on the moon, Mars, etc., there's these various different hazards that biology and life faces, right? Microgravity, ionizing radiation, also extreme atmospheres, extreme temperatures, elevated carbon dioxide, different circadian rhythm changes, reduced atmospheric pressures, as well as different confinement and isolation and behavioral issues that are connected to that, as well as hostile closed environments, which really alters the microbiome and other different health effects. A lot of these can also have combinations of multiple stressors, and so there's a long-standing field of science that's been undertaken, and we do have collaborators in Japan at the Japanese Space Agency, Japanese Exploration Agency as well. And so, yeah. So what I'm here to present to you today is a little bit on the Open Science Data Repository. And the Open Science Data Repository is really an integration of three NASA repositories. One is ALSDA. It's just an acronym that stands for the Ames Life Science Data Archive. And this was a 30-year-old database of imaging, bioimaging, radiation, O2, CO2, relative humidity, physiological measures, various different phenotypic, physiological, and behavioral type data, as well as an integration of NASA Gene Lab, which people may have heard of. This was developed in 2014 and was designed to really focus on omics and molecular data, as well as standardized pipelines to make the data comparable, and a tissue repository. There's a lot of different biospecimens and rodents and other different model organisms that go to space, and there's oftentimes surplus tissues that are available for request by the scientific community. And so together this forms OSDR, the Open Science Data Repository. This is a QR code to all the different tutorials, including the submission portal, which also has a curation portal to make the data maximally fair, as well as there's a file management transfer app for our submitters. There's a workspace for users and for labs and PIs that want to submit or curate or submit data and share data. We do have some human data, not NASA astronaut data, but there are other human data. So that's why we like to say it is maximally open, because we do have certain controls for private human data that is sensitive, although we do strive for it always to be maximally open. Our database, we're very proudly, is maximally fair. A lot of this was built off the history of NASA Gene Lab. And one of the benefits of having done all this activity to standardize data across our different repositories is that we've able to build these various different analytical visualization and even a knowledge graph tool, which should be coming online in the coming month or so or ish. And so that's all contained in that QR code there to the tutorials. NASA Open Science Data Repository, we have, it's small but mighty, I like to say. I think this also just because spaceflight samples are so precious and there's so few missions historically. But we do have really deep collaborations and connections, especially with the European Space Agency, the Japanese Space Agency, and we're starting to have more and more commercial entities submit data. Mostly we do have rodents, but we do have a significant amount of microbial data as well as human data, and more plant data. A lot of the different data in our repository is omics, largely transcriptomics, but we've recently added various different other types, from whole genome sequencing to metabolomics, you name it. And in 2021, and this was my position in the repository, we began standardizing the entire world of physiological, phenotypic, behavioral telemetry data. And up till now, we've now standardized 61 different assays across imaging to echocardiograms to tonometry, micro CT, Western blots, and we try to learn from experts, SMEs like yourself around the world to provide different standards for these physiological phenotypic standards. And we're very proud that there's been 93 publications around the world of scientists that have reused data coming out of our repository. So one of the reasons that how we've developed these standards is through our analysis working groups. So here's a snapshot. We have about 800 members around the world. It's open to the entire world. Anyone in the world is welcome to join. It's not organized by NASA. It's a community adjacent to NASA. And this is the breakdown of those 800 members. And so the AWG actually has two different purposes, this analysis working group. One is to help us determine the ingress standards of the various data, and one is the egress standards, especially like the AI/ML computational modeling SMEs. They really help us identify what's the best way for the output of the data as well. And for example, there was this notable package in Nature this past June where there were 44 articles across Nature portfolio, about a third of which came from analysis working group members working together. Because now that you've done all this work to standardize the data, you then, a lot of times this results in peer-reviewed publications, manuscripts being submitted. In terms of the standards for ingestion and reuse, getting more detail to why I'm speaking to you all today, we've sort of just are trying to standardize every piece of spaceflight, life science, health, biomedical related data. And so we're using something called the ESA-TAB model to standardize our metadata. We use this in terms of investigation, sample, assay metadata, and then the data files themselves. So we can deal with tabular results all the way to confocal microscopy, to MRI DICOMs, to Western blot gel images, right? So we sort of span the entire gamut, or to comprehensive metabolic panels from humans too. It sort of spans the gamut of various different data, but as long as we keep it in this structure, it helps provide this rich metadata. And we're very proud that our community has developed around the repository and has actually developed its own sort of community forum space where now anyone that's around the world that's interested that can get involved and find different open projects that are ongoing. Now, in terms of imaging, this is just a little bit of a snapshot of a couple examples of data sets. These were microbial biofilms that were studied on the International Space Station, and just an example of some confocal imaging. We also have like videos. These are actually videos of fruit flies that were on the International Space Station that were collected and curated, and we tried to work on standardizing the metadata as much as possible. Other examples of the imaging in our repository span from rodent research retinas to videos of the rodent research mice on the International Space Station, spatial transcriptomics, plants. We are getting more and more human data, but this is just a bit of a snapshot of our various different imaging and video that are available in the repository. And yeah, we also we live according to, we have to abide towards priorities and budgets. And of course, we're not right now going to have we're trying to learn from this community and your group here today. It could be the Bioimaging North America, it could be GBI, Qualrep Limi, and we're trying to, it's not NASA's place to reinvent the wheel. We're trying to learn from you all about the different types of imaging specific platforms. If there's how do we collect and curate, maybe it's labels on imaging most effective to make standardized machine learning pipelines, that type of thing. But we are proud of what we've done up till now, especially on the metadata side of things and ontology side of things. But we are collecting various different annotations of the data sets that are provided to us and trying to learn as much as possible from OME and especially the bioformats for our needs. We are already using this to a degree. And we do hope to expand and deepen our development of our database system for bioimaging in the future. So I really appreciate everyone's time today. Yeah, my name is Ryan. You're welcome to reach out. I do want to give thanks to my teammates and the entire community. I'm literally one voice amongst a amazing community of people that spend a great deal of time and passion and heart to put into this. From my management, but especially our hundreds of analysis working group members that have poured their hearts into the science and the work, and of course our stakeholders, the NASA Space Biology Program, the NASA Human Research Program, and others. So thank you very much.